Good afternoon, everybody, and thank you for the introduction, and thanks a lot to all the organizers for this nice event. And the project is about attentional, visual selective attention and attentional control. And just a very brief introduction for those who are not familiar with the basic concept of the attentional literature. Visual selective attention is the cognitive function allowing selection of relevant sensory input and the active suppression of interfering information. And there is a huge system in the brain controlling all the signals that can affect, in a sense, attentional control. And those signals come from very different cognitive domains and can contribute to a computation of attentional priority, which is then encoded in the so-called spatial priority maps. This is just a cartoon of a spatial priority maps, which is a neural representation in the brain, which is organized topographically and which represents space and all the sensory information within the external environment in terms of attentional priority. And attentional priority is, again, computed on the basis of very different signals. And for example, we have top-down attentional control, which refers to all signals related to the current goals of the individual. For example, if we are looking for a pair of scissors in a work table and the environment is very crowded, and we will use a template, an attentional template, a sort of model that we have, which is informed about what we know about the shape, the material, and the color of a typical pair of scissors. Then we have bottom-up attentional control, which refers, it's in a sense the opposite. It's an automatic process which is guided by the intrinsic perceptual properties of the sensory simulation, and in particular by the salience of a given stimulus. For example, here in the picture, you see the red apple grabs our attention because it's in a very homogeneous background of green apple. So it's a high contrast stimulus in terms of color. And the third category, which has been studied in terms of attentional control, pertains to a very heterogeneous signal that can be referred to as experience-dependent attentional control, which are all the signals that can derive from previous experience with given items, stimuli, locations. And so it's basically what we have learned about the environment. And one possible example here is that, for example, if we were exposed to rewards or punishments in previous encounters with a given stimulus, we will tend to shape our future attentional selection based on those experiences. And one other type of experience-dependent attentional control is the one based on statistical learning. So statistical learning refers to our ability to recognize, in a sense, capture all regularities in the environment and use them to guide our attention. For example, if we happened to find the scissors always in the rightmost part of the work table, we will go there with our attention first. Albeit we cannot, we might be unaware of this process. So it's an implicit shaping of priorities in the brain. All those signals have been longly studied, and we know a lot about the neural basis of all this type of signals. But what is the typical way to approach the study of those signals is in separate experimental context. And what is missing, and it's the general goal of the Macbrain project, is to understand how these different signals do interact in and contribute to the shaping of a unique attentional priority map and a unique computation of priority, which is the one that shapes our attentional choice in the real life, in everyday life. And so we try to understand, to unveil the degree of independence or interaction between different priority signals, and we consider the two extreme scenarios of having completely independent signals fueling and contributing to the computation of priorities, or at the opposite, those different priority biasing signals, attentional control signals, could interact and influence one another. And in the extreme case, one could take precedence in a given moment based on the needs of the individual or the context. And the study of attentional control signals and their interaction was done in relation to both target selection and distractor filtering in the Macbrain project. And the strategic plan of the project was basically organized in 3 phases, and the first 2 are almost done, and the third one needs help, in a sense, and e-brains can be one of the elements that would support this phase. And the first phase was to construct and validate a sort of reference paradigm, a standardized protocol to study all the different priority biasing signals in a unique experimental framework, so in the same context. And the second thing was to use multiple techniques to understand the interactions at multiple levels, from behavior to neurons, let's say. And the third thing is to synthesize, interpret, and model all the data. And this is the missing step for the moment. So the Macbrain Consortium was made of 4 units and representing different expertise. The unit in Verona, a unit at the Neuroscience Research Center in Lyon, and a unit at the University of Ghent, and finally a unit again in Lyon that's at the CNRS. And these 4 groups were basically involved in all phases of the project, but contributed with different kind of expertise. And so let's start from the paradigm development. What we did was to create and develop a reference paradigm, which was a very, very simple search task, and then insert different manipulations in the very same paradigm that can help testing different priority signals, either in isolation or combined. So this is the search task, and basically the participant had to look for the target, which was defined in terms of orientation. It was the bar tilted plus or minus 25 degrees of the vertical, while distractors were the ones tilted with the same angle but of the horizontal axis. And after selecting the target, the participant had to discriminate the location of the small gap inside the bar, so either up or down. And in the first phase of the paradigm, we had a cue stimulus, which in some experiments could be manipulated to investigate top-down attentional control, and it could be either valid, with one core indicated the upcoming target location with 100% validity, or neutral, which was completely uninformative about the upcoming target location. Then, after Q offset, the stimulus display appeared, and here we could manipulate different things. For example, in some of the trials, the display was homogeneous in terms of color, so there was the target and other non-salient distractors, but in some trials there was one of the distractor was colored in a different color, and this was a salient distractor. This was meant to engage also distractor suppression mechanisms, because this signal is very salient and you need to suppress it to go to the target, but also to study bottom-up attentional capture. And the third manipulation was about statistical learning, and we had also other ones, but I'm just giving you a few examples. And statistical learning was a manipulation of target frequency across the 4 locations. And so we had different percentage with which the target could appear in the different location, resulting in high, low, and intermediate frequency locations. And through this manipulation, we could obtain an implicit bias of attention in space towards the location which more frequently hosted the target, let's say. And those manipulations were tested both in isolation and then combined. We use the same paradigm because we think that it's really important that we evaluate interactions in a common scenario, because most of the experiments studying attentional control are optimized for a given priority signal, and this is potentially weakening the intervention of other signals. But in the same context, we probably have all signals that have the possibility to contribute equally. And in the first phase, we collected behavioral data. For example, we studied endogenous cueing in isolation, and as you can see here, we had a strong effect of the cueing, so reaction times were faster for validly cued targets with respect to targets with no information about the upcoming location of the target. But the interesting thing is here that we also have distractor absent and distractor present trials. And in distractor present trials, you can measure a cost of the bottom-up capture of attention by the salient distractor, but this was present only in neutral cued trials and not in valid cued trials, meaning that when the top guidance is strong, the bottom-up signal is gated, in a sense, in terms of behavior. And so it doesn't influence attentional control. And in this case, we manipulated instead statistical learning, and we had the typical effect. So reaction times were faster in the case of high target frequency location with respect to low target frequency location. In this case, however, the interaction was very different with the bottom-up signals. We again had the manipulation of about target distractor presence, but in this case, we had the main effect of the distractor presence, so the cost was found in all conditions, as if those two types of attentional control are acting independently, completely independently. They are not interacting in any sense. And so they contribute both, in a sense, to the deployment of attention. We then performed a combined experiment using both the endogenous cueing manipulation and the statistical learning manipulation. And the results are these ones. Basically, the main result is the interaction between the two signals. And what we see here is that we again find the effect of validity. So when the cue is valid, it helps target selection. And also, we found the general effect of statistical learning. But this effect is in reality only evident in trials with a neutral cue. So the difference in performance between high target frequency locations and the low target frequency location is only evident in the case of a neutral cueing. So when the top-down control is active and gives you the complete and valid information about the upcoming target location, you don't make use of information gathered about the location of the stimulus based on statistical learning. And these results were confirmed in another experiment, completely replicating the paradigm, but with the addition of the EEG measurements. And this is just to give you the picture of the behavioral results, which are identical. And in that experiment, we also measured brain activity and looked for many of the typical components that relate to attentional allocation in space. And I will show just one example of the results. So here is the computation of the N2PC component, which is a posterior lateralized component that is generally contralateral to the target location and is related to target selection. And here the raw data are shown for the ipsilateral and contralateral activity after the stimulus onset. And in blue you find the different waveform that subtracts contra-ipsi activity, which is the calculation for the N2PC component. And the N2PC is shown here. It's typically found here in this temporal window around from 200 to 300 milliseconds after stimulus onset. And here you see it is shown for the neutral Q condition, the valid Q condition, and for low target frequency location and high target frequency location. Just to sum up the results, we have here a graph showing the mean difference wave amplitudes for the 4 conditions. And what we see here is that a larger N2PC is found when the target follows a valid queuing, so we know about the location of the upcoming target. I'm sorry, but this effect interacts with the statistical learning effect, and there is an increase in the N2PC for validly queued targets only at low target frequency location. So here, this difference is huge. This means that in a case where we saw that from a behavioral point of view, the top-down attentional control gates, in a sense, the statistical learning effects, but in the brain, we have both. We still have both. So it's very different what the kind of interaction that happens in terms of behavior or what happens really in the brain. So even if the signals, for example, pertaining to some kind of attentional controls are not evident in the final attentional choice, in the final behavior, they are there somewhere in the brain, ready for maybe to act in case of need. And we also measured fMRI activations in many different, for many different signals. And for example, we studied the combined influence of top-down attentional control and salience. And in this case, we used only valid queuing for different quadrants, and 2 and 3 conditions in terms of salience. So we had the homogeneous display, a display in which the target was salient, and a display with a salient disruptor. And what we measured was, first of all, the activation, the quadrant-specific activation in the visual system, in the visual cortical areas. And I will not go through the details of the computation, but basically what we found was that the brain activation in quadrant-specific area in the occipital pole was modulated by the allocation of attention in the different quadrants. And so what was modulated, it was modulated by the top-down attentional guidance. And with we computed a metric describing the orientation of attention in space and its strength, which was coded as a spatial bias vector. and we realigned all the quadrants just to have the target quadrant, the distractor quadrant, and the other quadrants. And we computed all the spatial vectors in all conditions. And what we found was that the spatial bias vector, so the direction of attention, is obviously under the influence of top-down guidance, which is reported as the vector here, that goes towards the target in quadrant, which is depicted in black. But this vector is modulated by the presence of a saliency related signal. And basically, the length of the vector here represents the amplitude of the bias. And the bias is reduced in the case there is a salient distractor, and is enhanced in the case there is a salient target. So if the two attentional control signal go in the same direction in terms of what is good to be attended, they will had their benefits, in a sense. Otherwise, they will will have different. The two signals will be addressed to different locations. In one case, the target. In the other case, the distractor. So the distractor-related signal will diminish the effectiveness with which attention is directed to the target. So this is just, we also did dynamic causal modeling, trying to understand connections between the quadrant-specific activations in the occipital cortex, and how these activations were related to areas that are known to contribute to attentional control and to host spatial priority maps, as the PPC. And in this case, the modeling was selected, trying to compare different possible architectures, including feed-forward, feed-back connections with all quadrants or only with relevant quadrants. And all the models were compared, and in the end, what was the general picture is that both lateral connections within the occipital cortex and connections with the parietal lobe contribute both to top-down and bottom-up, the encoding of both top-down and bottom-up control signals. And also the strength with which different connections are shaped and modulated by these different signals is modulated by the role that the salience-related signals has in the computation. So if salience is the salience of the target, it contributes to the strengthening of some of the connection. For example, the one linking the posterior parietal cortex with the quadrant encoding the target. But then, if you have a signal, a saliency signal, which is related to the distractor, then this changes the lateral connections within the occipital cortex, and in particular, it acts on the competition between the two relevant quadrant, the one hosting the target and the one hosting the distractor. And this was just a brief overview of the type of experiments that we were conducting in the Macbrain project. These are the main techniques that were used in humans. Then we have a whole story, which is not shown here, that is related to electrophysiology in the non-human primates. And this is not here because the data are still being collected and is not finalized. But the aim of that part is to have the very same, the identical behavioral paradigm also for the monkeys. and to insert minor adjustments to have those data to be fully comparable with those collected in humans. And so, to sum up the main scientific results, we obtained clear, reliable effects of all the attentional control signals we tested when presented in isolation. And we found an overall predominance of the top-down attentional control over other signals when they were active together. However, this prevalence of the top-down attentional control was very different in terms of behavior and in terms of brain activation. So it exerted a sort of a gating effect on the influence of other signals in terms of behavior, but the other signals were still contributing to the brain processing of all the information present in the task. And so we also gathered some information about the interaction between different signals in the brain, also biomodulation of the affective connectivity between different areas involved. And to get a broader picture, the most important thing that we can highlight in the project in terms of general achievement is the development of this reference protocol, which is suitable for investigating the influence of very different type of attentional signal within a unified experimental framework, and that can be applied both to human and non-human primates, and also used with very different techniques. And we collected data which are already a good data set pertaining the neural integration and the behavioral integration between these different signals, and we are still trying to enrich the data set with monkey data, which are again ongoing. And so we have some behavioral data, but the implementation of the task was not so simple for the monkey, because when it comes to combine multiple signals, the training is a bit difficult, and so we had to make some adjustments, but we want to have them to be really minor to have the data to be comparable, so it's not so easy. And finally, we started the data sharing process, but as it was said this morning in other talks, we didn't start with the e-brains. We didn't know that the possibility to share the data was so easy and so useful in a sense. So we added the general, we took at the beginning the general decision that the data will only be shared after publication with the scientific community, and but the interesting thing of e-brains, I think, is that it offers many different ways to share the data, and you can use it also for internally sharing in a group. And also, we really appreciated how difficult it is to share the data with e-brains. I'm not joking, is because they really want you to organize the data in a way that is understandable for many other people. And when you put the data in a public repository, normally you use your way. And you just put the data there. They are available, but they are not so clear for people working in other domains. But eBrain stressed the fact that the format should be homogeneous, and this is good, I think. It's an effort because you are not immediately ready to share the data, but then it's something better for really sharing and not, yeah. So thank you. This is it.